Hello everybody, so today we are going to discuss about the Basidiomycetes, Basidiomycota. So Basidiomycota basically, previously we are known about Ascomycetes. What are Ascomycota? Because of their special sac-like bladder-like structure known as asci. Here Basidiomycetes are famous because of their structure, which is road-like, or in other words, it is club-like structure, road-like structure, which is known as Basidia. Basidia plural, Basidium singular, and this is also the reason we're calling them as called club fungi, because they have club-like Basidium structure, right? After that, when it comes to their structure, they do have sexual reproduction as well as asexual reproduction, but asexual reproduction is quite uncommon, which means asexual reproduction uh, does not occur in many cases, but occasionally if it occurs, then it occurs by the help of formation of conidiospores, right? And here, when it comes to its hyphae, in in Zygomycota, we basically had a septate. In Ascomycota, we had septate, and here Basidiomycota also has septate hyphae, xenocytic, non-xenocytic or uninucleated hyphae, right? And they have septa crosswalks present between them. Moreover, they have different types of hyphae present, such as one of them is conidiospore, right? One of them is conidiospore. uh, Spores, sorry, not spores. Spores are the hyphae inside which there's conidia, which are the you know uh, structures in which the conidiospores are actually present, right? After that, when it comes to some other structures, um, they usually form a fruiting body, a large network of fruiting body, which is known as the basidiocarp, and that basidiocarp is also shown on your screen here. That here you can see the basidiocarp present over here. This is what we call as a basidiocarp. And here, when it comes to uh, the structure is in such a manner that here, the basidia basically it is a sexually reproductive structure. Sorry, sorry. Here we are having basidia. Let me just take out my pen. All right. So when it comes to the structure, they are having, you know, a sexual reproduction, after which they convert into asexual reproduction, which means here, first there is formation of, a, their life cycle all starts. We are only considering over here the life cycle of Agaricus. Because Agaricus is a species of Basidiomycota phylum, and once we consider a single organism, it is an example for all other organisms, right? So here, uh, when it comes to discussing their structure is, first and foremost, they basically have development of basidium. That basidium developed is heterokaryotic. It is heterokaryotic basidium, which means basidium is having n plus n nuclei, which means it is having two nuclei, and both of these are separately present within the same cell, and that is why we call this n+n arrangement, right? N+n arrangement is there. And apart from that, when it comes to their structure, basidium is arranged in such a way it is a rod-like structure. It is just like a rod-like structure, and that rod-like structure basically has four uh, stalks or finger-like projections, which are known as four finger-like projections are there. Four finger-like projections are there. These four finger-like projections are known as spermangia, and these spermangia, each of the spermangia is having a spore, which is known as the basidiospore. Each of the spermangia is having a spore, which is known as a basidiospore, which means each basidium is having four basidiospores over it. Now, these four basidiospores are basically what? These four basidiospores are haploid, which means meiosis occurs in basidium. As a result of which, when these are haploid, these spores, when they disperse by the help of wind, they are fertilized together, and as a result of this fertilization, it causes to produce a structure which is known as, when they fertilize together. One more thing, actually, these are diploid. Yes, these are diploid, and from there, they have asexual reproduction occurring. Through the help of asexual reproduction, they cause to produce their hyphae, and asexual reproduction occurs by means of nuclear mitosis. As a result of nuclear mitosis, they tend to develop their hyphae, which is again heterokaryotic, due to which they have a delay in their because there's a delay in their heterogamy and plasmogamy, due to which they have a delay in the heterogamy and plasmogamy, due to which they have an heterokaryotic stage, which starts from here by the formation of the hyphae, in which there is haploid hyphae formation. From there is formation till the time there is again development of the basidium, and from basidium the diploid zygote. There is an heterokaryotic stage occurring, which means it is a lengthy heterokaryotic stage. And which means our secondary mycelium is present in most of its life cycle, and primary mycelium is present in less of its life cycle. Likewise, when it comes to some other characteristics, it is having monokaryotic stage is less in its life cycle, and heterokaryotic stage is more present, or dikaryotic stage is present more. Right now, this is how the structure is developed. So structure here, when it comes to its structure, here we know that haploid nuclei is present and meiosis occurs. Haploid nuclei is present and meiosis occurs, right? So when meiosis occur now here, we were on discussing about the primary mycelium, right? So from primary mycelium there is development of secondary mycelium. Secondary mycelium is just simply one which is having the dikaryotic stage. Dikaryotic stage represents the secondary mycelium. Monokaryotic stage is associated with primary mycelium. So from the secondary mycelium they develop several structures. From the secondary mycelium, uh, they develop several structures which are known as the they develop structure which is known as from the secondary mycelium they develop a structure which is known as a basidium, and from the basidium they develop a structure which is known as basidiospore, and from the basidiospore they develop primary mycelium, and from the primary mycelium they develop the secondary mycelium, and from the secondary mycelium they develop a structure which is known as button. Button, basically it is nothing special, but the button is nothing special but a specialized structure from where the fruiting body basidiocarp will arise, or in other words it is the network of several basi in other words it is basically we can in simple words we can call it as a network of basidia. Network of basidia which contains multiple basidiospores is known as button. Network of basidia which contains multiple basidiospores is known as a button, such that this network of basidia having multiple basidiospores is known as button, from where the fruiting body arises, and that fruiting body arises is known as basidiocarp. And that fruiting body has a separate anatomy of it, that how the fruiting body looks like. So when the fruiting body arises, the fruiting body has got a stalk. This is a stalk, or in other words you can also call it a stem or stipe. And apart from that, down towards it there are mycelial threads are present over here as well. Apart from which, when it comes to its upper part, this upper region is known as the cap. Upper region is known as cap. Some of the species are also having scales over the cap, but some also are not having it. And on the other hand side, some are also having a ring like structure present on their stem, which is known as ring of annulus. Apart from which, when it comes to the cap, down towards the cap, on the interior surface of the cap there are several thin perpendicular plates. There are thin perpendicular uh, plates, on the inner structure. Let's suppose this is an inner structure, and from this inner structure there is radiation of several thin perpendicular plates which start from the end of the stalk and go along the edge of the cap, which is known as the gills, or in other words gill plates as well. And these gill plates are the sites from where on the tip of these gill plates we are having the structures which are known as on the side tips of gill plates we are having the structures which are known as the basidiospores, which are basically the haploid ones, haploid spores. And these haploid basidiospores, when these are germinated, they develop into a structure which is known as basidium. And once the structure basidio of basidiospore is developed, uh, basically when it comes to basidiospore, particularly two basidiospore here, they fuse together. They fuse together. Two basidiospore fuse, due to which plasmogamy occurs. After plasmogamy, the heterogamy is stopped at a while. Once a plasmogamy occurs, heterogamy is delayed, due to which heterokaryotic stage occurs. After which, when the two nuclei are then again fused together, they cause to produce a structure which is known as the diploid zygote. And that diploid zygote now undergoes meiosis, as a result of which it causes to produce a structure which is known as basidium. We know that meiosis produces four haploid cells, due to which four haploid cells are produced on the from the structure of diploid zygote, such that diploid zygote now is converted into a rod-like structure which is known as basidium, on the tip of which there are four stalks known as spermangia, and over each stalk spermangium there is one particular basidiospore, and that basidiospore is haploid in number, right? And once it is haploid in number, that haploid basidiospore will now again tend to separate by the help of dispersion through wind. And as a result of the dispersion through wind, it will tend to produce a primary mycelium first and foremost, because nuclear mitosis, because here heterogamy and plasmogamy will occur first, after which secondary mycelium will develop after primary mycelium, in which nuclear mitosis will involve heterogamy as well as plasmogamy, right? Now again, from the secondary mycelium, button will be developed, a network of basidiospores having basidia arranged within it, from where again there will be development of the basidiocarp, and from basidiocarp there will be dispersion of basidium from the gills, tip of the gills, due to which again there will be fusion of the two basidia and two basidiospores, as a result of which there will be plasmogamy occurring first, then heterogamy will occur after some time, as a result of which there will be a delay, and heterokaryotic stage will persist. As a result of that heterokaryotic stage being persisted, this will lead to the fusion of the gametes, or the nuclear fusion, or the karyogamy after a period, due to which when nuclear fusion or karyogamy will occur, it will cause to produce a diploid zygote, due to which the cycle will again occur, right? So this is how actually the cycle occurs. And some common examples of basidiospores includes examples of Puccinia, smut and rust species. Smut example is Puccinia, and rust species example is Eustilago. These are basically parasitic. They tend to, you know, they are dependent on their host. Rust is an obligate parasite, right? And example includes Eustilago and Eustilago metis. Eustilago triticus and Eustilago metis, right? Eustilago triticus, Eustilago metis, right? And apart from that, Eustilago triticus attacks on wheat, Eustilago metis attacks on corn kernel. And how do they actually occur is that they are named as rust and smuts. Rust basically resembles the rust-like structure. Why are these named as rust? Because they produce, you know, several red brownish, um, rust-like spots over their host plant. For example, most of the time they attack over the stem and leaves. So in that case, they cause several rust-like spots, such as reddish or brownish spots, which cause to give them a rusty appearance, such that there has been a rust over that particular plant, but in actual that is the rust species of fungi which has caused that damage to that particular crop, as a result of which these are known as rusts. While when it comes to smuts are named because basically Puccinia is a rust species, and Eustylagotriticus and Eustylagomedes are the smut species, right? And smuts are basically known as uh, known because they produce dark, powdery, dusty, smut-like spores in the crops of this particular plant. Let's suppose if we take the example of wheat. So when it comes to wheat, wheat has several, its cereal grains are present over it. Cereal grains, let's suppose there are several cereal grains present here over it in the wheat crop. So they basically cause to replace these cereal crops, and instead of these cereal crops growing, they tend to produce their own spores, which are smut like, dark black, dusty spores, which tend to grow instead of the kernel crops and ultimately destroy the entire crop. So as a result of this, these are known as smuts because they cause to produce smut spores, which tend to destroy the entire crop. And same goes with same goes with z maize, the corn maize plant, such that it is also having the same case that its grains are replaced by the Eustylago amidis spores, right? And there's a complete life cycle for it, such that when it comes to their life cycle, when their spores are found on a particular plant. Let's suppose if we take the example of Eustylago triticus, when their spores are found on a particular plant, these spores are dispersed by wind to another plant, another crop. Let's suppose this is one wheat crop, this is one wheat crop, and I'm drawing another one. There's another wheat crop, right? This is another wheat crop, and from that wheat crop, these spores are being dispersed through wind or any other source, most of the time through wind. So from that source, these are then transferred to another wheat crop, and once these are transferred to that particular wheat crop, it leads to transfer these spores into the flower of that wheat crop. Let's suppose I'm just trying to draw a rough structure over here. uh, Oops, I'm just trying to draw a rough structure. uh, you know they tend to go into the flower of the wheat crops, and once they go into the flower of the wheat crop, they ultimately develop their mycelium into the ovary of the wheat crop. They develop mycelium into the ovary of the wheat crop. Mycelium are developed. Mycelium is developed in the ovary of the wheat crop. Now, once mycelium is developed, that mycelium being developed causes to produce its hyphae further into the seed or grain of the particular seed, or it causes to develop its hyphae into the particular seed or grain of that particular ovary, as a result of which, when that particular seed is germinated the next year, when it is sown into a new crop the next year, it causes to produce it causes to produce as a result of which it causes to produce a new crop, alongside which, when the new crop is grown, the growth of that particular smut, Eustylago triticus, also takes place, such that its hyphae are also grown alongside the growth of the crop, as a result of which that hyphae causes to produce smut spores inside the crop, replacing the grains of the crop, ultimately causes to destroy the entire crop as a result of which this causes to destroy the entire crop, and the crop is ultimately destroyed. And again the cycle starts from where it ended, such that again these crops are dispersed by wind and go through our to other plants, and again go into their flower, then ovary, then develop hyphae into the seed, and then the seed causes to again germinate, causing the growth of the hyphae alongside the growth of that crop, due to which again there's replacement of the seeds by the particular smart spores of that particular fungi, as a result of which that plant is damaged. So this is how the process actually occurs. Some common examples of Basidiomycetes are also also it includes examples of some edible fungi, uh, mushroom. Agaricus, there are 200 different edible species of mushroom. So example of this includes Agaricus bisporus, most commercially utilized species of fungi, which a species of mushroom which we utilize as a source of food, as well as there are other species such as compestris, Agaricus compestris as well, and apart from that, we are also having another species. Let's take the example of Agaricus compestris, Agaricus bisporus. Some poisonous fungi are there: Amanita, jack-o'-lantern. These are the poisonous fungi. We are going to discuss about them in detail in the next class of importance of fungi. Moreover, when it comes to deuteromycetes are basically, these are the most imperfect fungi. These are named as imperfect fungi on the basis of their most uncommon characteristic, which is that they do not have any sort of sexual reproduction. Sexual reproduction is not observed. These basically were associated with different phyla, such as most of them were associated with Ascomycota, while some of them were also associated with Basidiomycota and Zygomycota. But since they did not show any sexual reproduction, they were then taken out of these phyla and then placed into a separate phyla, which was a phylum which is known as the Deuteromycetes, and that Deuteromycetes is known as the fungi imperfecti, which means it includes all of the fungi which are not having sexual reproduction. All are imperfect, right? And apart from that, when it comes to their structure, they can be septate as well as aseptate and aseptate. Most of these, it depends on the particular phyla from which they are taken. As we take the example of penicillium, penicillium is was previously a part of Ascomycota, due to which it is considered as septate. And uh, when it was observed that there is no sexual reproduction occurring in penicillium, it was then kept in Deuteromycetes, and here it is again having the same hyphae, which is septate. And apart from that, when it comes to their sexual reproduction is not observed, but asexual reproduction is observed, which is which occurs through conidiospores, as well as their they most of the time they have development of conidiospores, while some also undergo fragmentation, right? And apart from that, when it comes to the structure of penicillium, uh, first, when it comes to some examples, Aspergillus, most famous species, Aspergillus penicillium species, and Neurospora, as well as there is Fusarium, Helminthosporium, as well as Alternaria. These are the famous species of Deuteromycetes. After that, uh, Neurospora, it is commonly known as pink bread mold, pink bread mold, and Aspergillus brown mold. If I'm not wrong, it is brown mold. And apart from that, when it comes to penicillium, when it comes to penicillium, it has a common name which is blue green mold. All of these fungi basically are having a characteristic color, such as penicillium having blue green, Aspergillus brown, and Neurospora giving their characteristic pink color. This color is basically due to the color of their conidia. Their conidia are having specific color. Let's suppose if we take penicillium, they are having specific color of their conidia as blue and green, due to which they show a bluish green color of their entire body. So their entire body's color is dependent on their conidia, and their structure is developed in such a manner. So here we are having diagram of penicillium. They are found in mycelial colony. This mycelial colony is circular in manner when it is appeared, and here it is having a vegetative hyphae from where there is development of conidiophore, and that conidiophore is a single stalk-like structure. From there is a development of several networks of several networks of stalks, and these stalks are developed in such a manner. This stalk which is developed is known as medullae, and from this medullae there is arrangement of several conidia which are in the form of network like arrangement. First, the conidiophore is developed into a medullae, and that medullae is further networked or branched into sterigmata or fialets, and these sterigmata and fialets individually contain a chain of conidia, one on the top of another, as a result of which this causes to produce a network like arrangement. And the mycelial colony, as a result of this network like arrangement, appears circular. And these conidia are important, uh, because of their characteristic color, as well as these are circular in shape. And moreover, when it comes to their conidia are, they have certain characteristics of conidia. uh, you know they contain the conidiospores, which are easily dispersed by the wind, very easily. And apart from that, when it comes to some other structures, here we are also having the diagram, only it is a bit in detail, such that the conidiospores are developed, these are branched into several structures which are known as stipe. Stipe is further branched into rami, which is further branched into ramuli, which is further branched into medullae, and medullae further branched into fialates, and fialates further finally having the conidia top of one another on the fialates or stermigeta. So in this case, this is how the arrangement occurs, and this is how we actually classify. This is all about this video. Hopefully we'll meet again in another video.